Thank you, Jan, and thank you to the Sutter team for inviting me here today. I'm very excited to talk about my work using dynamic clamp to explore how sodium channel gating regulates Purkinje neuron firing. And I wanted to start off with just a brief introduction to the cerebellum. And the cerebellum is classically involved in motor coordination, where it takes input from sensory information, integrates that information, and then sends the appropriate output. And this is just an illustration of a sagittal view of a mouse brain. And here we have the cerebellum kind of on top of the midbrain here. And if we take a look at one of these structures inside the cerebellum, this is called a cerebellar lobule. And we can zoom into this and we can see what's called the cerebellar cortex. And the cerebellar cortex involves a bunch of different neurons that regulate the output of the cerebellum. And I want you to pay particular attention to these green cells, and these are called Purkinje neurons. And Purkinje neurons are very important to the cerebellar cortex because they are the sole output of the cortex. And these neurons have the intrinsic capacity to fire spontaneously at very high frequencies, anywhere from 50 to 100 times a second. And this output is very important for cerebellar function, primarily because they are the sole output of the cerebellar cortex. And this high frequency firing is primarily mediated through sodium currents, which is facilitated through proteins in the membrane called voltage-gated sodium channels. And here I have a sodium current, a modeled sodium current based off of a Purkinje neuron current. And I just wanted to go through all of the different current components that we can see here. So if we step our cell from a resting potential, such as -90, and we depolarize the membrane, the voltage-gated sodium channels will activate and open and allow for sodium conductance. They will very quickly inactivate. And this is what we call the transient sodium currents, or INAT. And so this is called fast inactivation or conventional inactivation. And that's what contributes to this transient sodium current. And if we keep this depolarization for a couple milliseconds, maybe 5 milliseconds in this case, and then we step back down to an intermediate voltage, such as -45 millivolts or -50 millivolts, some proportion of the channels that have undergone fast inactivation recover from fast inactivation and contribute to something called the resurgent sodium current, or INAR. And INAR is not as big as the transient current, but somehow these channels are able to recover from fast inactivation and open back up to allow current flow through the membrane. And then this resurgent current, as you can see, kind of decays over time. And it doesn't go back to baseline. And this is due to something called the persistent sodium current, or INAP. And INAP is contributed by channels that are not inactivating or are slowly inactivating and remain open and allow for sodium influx during this depolarization. And it's important to note that the resurgent current is thought to underlie this high frequency firing that we see in our Purkinje neurons. And I kind of want to shift a little bit to dynamic clamp that we can use through Sutter Patch. And I can't really talk about dynamic clamp without talking about computational models, right? So this is our Markov state model of a voltage-gated sodium current. And each of these states represent the conformational changes that you would typically see in a voltage-gated sodium channel. So this is a closed state. Here is our open and conducting state. And then you have these transition rate constants in between each of these states that determine the math that goes between each of these states. And I wanted to focus in on 2 particular states, one of which is this fast inactivated state, which, as you remember from the previous slide, occurs after the transient current. So this is a very fast inactivation where it terminates that transient current and we get that really sharp termination of that current. And so while some of those channels can go from the open conformation into the fast inactivated state, other channels can go into what is called the slow inactivated state, or the IS in this case in this model. And if we depolarize the membrane enough, while some of those channels will go into fast inactivation, some of them will go into slow inactivation and remain open, but over time, those channels will not conventionally close, like the fast inactivation state, but will kind of collapse in on themselves, which terminates the conductance. So all of this to say is that we can use this Markov model to readily recapitulate all of the different current components that I mentioned previously. And we can do this through Sutter Patch and through Dynamic Clamp. So we can model this current on our computer, and then we can apply this current to real cells by connecting our software to an amplifier, which provides the current for Dynamic Clamp. And so if we're patched into a neuron, for example, a mouse cerebellar slice here, and we're patched into a Purkinje neuron, we're recording in voltage clamp, right? Or sorry, we're recording in current clamp, we're recording the voltage changes, the action potentials, the mathematical operations within Sutter Patch and within the model will determine the appropriate amount of dynamic clamp mediated current to be added afterwards. And that's what that looks like here. So on top in black, we have our action potentials that we're recording from these real neurons. And then in red, this is the dynamic clamp mediated current that's being produced by this nominal model, our baseline model. And after we add this current, this dynamic clamp current, what we see is a significant increase in firing frequency, which isn't all that surprising because we're adding a depolarizing current on top of cells that already have this current. So we're just increasing the current, which increases the firing frequency on top of our cells. But this allows us to really look into how firing frequency changes if we change properties of our model, which then changes properties of the current, right? So our first question was, what happens to Purkinje neuron firing if we want to scale the resurgent current in our model? And remember, we can simulate these currents by producing this nominal model with the transient, resurgent, and persistent components. But if we change and manipulate those rate constants in between the states, we can then create new models that change these current properties. So for example, we created 2 additional models, one of which decreased the resurgent current by about 63% comparatively to our baseline nominal model black trace here. We also created a model that increased the resurgent current by nearly 250%. So we have all 3 of these models. We can then add our models through dynamic clamp in Sutter Patch while we're patched onto real cells, and then assess firing frequency changes as we add the nominal, the increased resurgent current, and the decreased resurgent current models. And when we do that, we can then compare all of the firing frequencies that we see after we apply those models. So here we have our baseline nominal model firing frequency in our cells. After we added our resurgent current decreased model, that's this red trace here, what we found was a significant reduction in firing frequency, which isn't all that surprising, primarily because the literature tells us that the resurgent current kind of drives this spontaneous firing frequency. So a model that's added with a reduced amount of resurgent current, seeing a reduced amount of firing frequency is not that surprising. However, when we compared our increased resurgent current model with nearly 250% increased resurgent current compared to our baseline, we didn't see any significant differences, which really perplexed us. And so we really wanted to see how we can manipulate our models further to really break down what is exactly happening with this result here. So our next strategy was to develop models that vary not only in the proportion of the resurgent current, but also in the proportion of the persistent current. So in developing these models, we asked ourselves 2 questions. How does more resurgent current impact firing? But also, how does more persistent current impact firing? We can then apply those models to real neurons using dynamic clamp, and then we can assess how scaling the resurgent and persistent currents in those models affects the repetitive firing of those cells after we've added those models in through dynamic clamp. So we have our recordings of our cells that we have added our different models to it. And then we can compare it, we can compare those firing frequencies to the current properties of our simulated currents of each of those models. So we can test against the amount of resurgent current in those models, and then we can test the amount of persistent current in those models. And so here's a graph of the percent increase in firing frequency on the y-axis. So as we get higher, we have a greater increase in firing frequency in our cells where we applied the models. And on the x-axis, we have the amount of resurgent current within those models themselves. And what we see with this comparison, this is the resurgent current here on the x-axis. even if we add a large amount of resurgent current, we're not really seeing a correlation between the amount of resurgent current added to the increase in firing frequency. And that's especially evident by our correlation coefficient R squared, which is quite low here. However, when we compared the persistent current in these models against the percent increase in firing frequency, we saw a strong positive correlation between the amount of persistent current and the firing frequency of these cells, suggesting that the more persistent current that is in these models, the greater the firing frequency. And you can see our correlation coefficient is much greater here. So this tells us two things: that the resurgent current does not necessarily scale firing, and that also the persistent current actually reliably scales firing frequencies. So that still leaves us with one more question to answer, right? And that's, what is the resurgent current's role exactly in Purkinje neurons? Because it's a really interesting current, right? And so we're going back to our Markov model just briefly, and we wanted to focus in on this slow inactivated state, this transition between the open and the slow inactivated state that is mediated through this rate constant A2S. And remember, we can create our simulated currents. This is our nominal model. And we wanted to decrease the transition rate constant that goes from open to slow inactivated state. So the model will spend more time in this open state. So we're decreasing A2S. And what happens when we do that is that we have an increased persistent current, but a decreased resurgent current. And then if we plot this over multiple models, as we decrease our A2S value, this transition rate constant going from the open to the slow inactivated state, we see this reciprocal relationship between the peak resurgent current on the x-axis here and on this z-axis here against persistent current. So we see this correlation between, this reciprocal relationship between the resurgent current and the persistent current. And if we add this new model where we've decreased this transition rate constant through dynamic clamp to our neurons, and we compare it against this baseline nominal current, we see a significant increase in firing frequency. So what does that tell us? That tells us that the persistent current is the one that reliably scales Purkinje neuron firing frequency. And it's the resurgent current that may act as a reservoir of sodium channels that are slowly inactivating, which contributes to the persistent current, and it is the persistent current that scales the Purkinje neuron firing rates. And with that, I would like to thank my advisor, Joey Ransdell, as well as our collaborators at Washington University, the Miami University Animal Facility, as well as the guys at Sutter Instrument, especially Telly, who has listened to a lot of my emails. Thank you so much.